Alright, hello, Assalamualaikum warahmatullah. So we are going to move on to database mining, which is how are you going to look for information on the internet on if let's say you have a sequence, so what is the next step that you need to do. Okay, so if you go to the internet, there is a website called NCBI, okay, National Center for Biotechnology Information. So this website tells you it's basically a bank of storage bank for genes, proteins, all those sequences. That is, everything is dumped in here. So you can submit and download, learn, develop, analyze, and do research based on the sequence that you have. And if let's say you can have popular resources right here. PubMed is the journals and articles that you can use to refer to refer to. And you also have BLAST, which if let's say you have just received your sequence from let's say you receive from the email, let's say you have sent your plasmid sequence to a company, and you ordered them to sequence your gene. So you gave them your plasmid and they sequence it for you, and the sequence came back to you. And when the sequence came back, it looks something like this. Alright, so they're gonna give you this. Let's say so please download your required DNA sequencing results. So you have all this blah blah blah, and they and then let's say it's gonna come out something like this. Okay, the DNA sequence. If let's say they give you in that, after you have downloaded your sequence, you saw this. And the next thing you need to do is copy your gene, and you go to BLAST. Okay, BLAST. So this BLAST is something that you can find on NCBI. Okay, so right here, okay, BLAST. Okay, so you have nucleotide BLAST, BLASTX, T-BLAST, and protein BLAST. So because we have a nucleotide sequence, so what you need to do is choose nucleotide BLAST, because it helps you to find which ones that is most similar to you. But if let's say you want it to be translated into protein, so you choose BLASTX. But if you have a protein sequence, you want it to be reversed back and translated into nucleotide, you use T-BLAST-N, and probably that's not that accurate, because some of the protein has different and if you have protein, that is you can use protein BLAST. Okay, so since you have nucleotide to nucleotide, so click on that and paste your sequence and click BLAST. And right here you say you want it to be highly similar sequence. Okay, so you click BLAST. It's going to take a while because they're searching the result for you. Okay, and it came out something like this. You have 3 sequences, okay, and max score, total score, your cover. So a percentage of identity is 100%. That means it is the thing that you are looking for. And the E value means okay, the E value is actually okay. So the E value here refers to, they try to, it's an expected expect value, which describes a random background noise. If let's say sometimes you have unclean sequence, so they help to reduce all those background noise. So something like that. So if you have so much noise, it's not clean, so it's hard for you to find identical sequence. So since this one is probably it's clean, and so that's why you can get a percentage of identity up to 100%. Okay, so that is for sequence and blasting. If let's say there's a question that asks you okay, go to this website again. If let's say I am looking for a nucleotide, I don't know, probably okay, I'm just gonna use strictosylene. This is my gene I was working on with when I was doing my master. Okay, so on this side, results by taxon, sorry, nothing right here, but usually there is something here which tells you that there is a tree. Oh yeah, there is a tree right here. Okay. Sorry. And probably okay, so a tree, it's something like this. Okay, choose cladogram. Okay, so a cladogram, you probably have learned this in school, but probably not that detailed. A cladogram tells you if, let's say, the percentage of identity is 99% or 88%, and then there are also 50, 100%. So the ones that have the closest identity will be on the same plate. And the furthest away, probably this, let's say it's 15%. It's gonna sit outside, which is called the outgroup. Oh, yeah, outgroup. Or probably this branch right here, it goes out. So that's what we call outgroup. Okay, so the ones that have the most similarity will be on the same plate or branch right here. All right. Okay, so that's how we do it. So that's for a start for today. So if you have any question, you can just ask me, and we're gonna go